All right, in section 22.4, we're going to talk about how DNA replicates itself. It's basically what happens is you have a parent DNA, which we talked about in the previous slide. You have a parent DNA that helps produce our daughter DNA, and the daughter DNA is the opposite of the parent. Okay, so if you have an A on the parent, it actually makes a T in the daughter. And basically what happens is you have your strand unwind itself, right? It unwinds. And what happens is our 3 to 5 prime, okay, strand replicates itself in the opposite way, which is basically the copy of the 5 to 3 strand. Okay, and then the 5 to 3 strand makes a copy, and it's the equivalent of the 3 to 5 strand. So it makes the opposite of itself. All right, and I'll kind of show you what that means. It's called a replication fork because I guess it looks like a fork. It starts unwinding and it starts to get replicated as it's unwinding. So let's kind of look at a blow up. Okay, so you have all of the nucleotides here. All right, and basically what happens is here's our parent. This is our parent strand, and then you have an enzyme which is called DNA polymerase. All right, and what it basically does is that if I have an A here, it brings over a T, because remember A and T go together. So it brings a T over, and it brings a T over. And if I have, I don't know, let's say I have a G here, it brings a C over. If I have a C here, it brings a G over. So it's not replicating each strand identically. What it's doing is it's making, so if you notice, these base pairs is the equivalent of these base pairs on this side. So pretty much this is being replicated, and it's going to be the exact same as this side of the strand. And then the opposite is true. Whatever is on my parent here, the opposite is being replicated. All right, so you can see that these nucleic acids are the same as the nucleic acids over, let me see if I have it right. It looks like it's these right here. It's the identical over there. So this parent strand is making the daughter strand, and this daughter strand is the identical of the other strand on the DNA, and then this strand right here is the exact duplicate of that. So it's always making the opposite. All right, and it goes from the 3 to 5 direction. You have these terms, what's called a lagging segment versus a leading segment. A leading segment is that huge strand that's constantly being made. Okay, a lagging is just a small little portion. Eventually, this small portion will hook up onto this portion, and you'll just have one long leading strand. Okay, so they talk about it here in replication. So the identity of the bases of the template strand determines the order of the bases on the new strand, and it's always the opposite. All right, so A must pair with T, G must pair with C. The new phosphodiester bond is formed between the 5-phosphate and the 3, which we've already learned. There's nothing new there. Replication occurs in only one direction. It goes from the 3 to the 5, the 3 end to the 5 end. And the new strand is either leading or lagging, meaning it's growing continuously, which is leading, or lagging, it's growing in small fragments. Okay? So now let's talk about RNA, because RNA is really responsible for making our proteins. Okay? So there are a few differences to RNA, which we've talked about. One, the monosaccharide is ribose. It's not deoxyribose, so that's kind of the big deal. The other one is that it doesn't use T, it uses U instead. So rather than having an A to T form, you have an A to U form. All right, RNA is a single strand and a smaller DNA. It's not a double helix, it's a single strand. Okay, and there are 3 types of RNA which we will quickly go over. There's rRNA, which is ribosomal RNA, mRNA, which is messenger RNA, and finally tRNA, which is transfer RNA. So ribosomal RNA provides the site where polypeptides are assembled during protein synthesis in the ribosomes. So RNA is really, it takes the information from DNA, and it's actually responsible for synthesizing our proteins. Okay, every 3 nucleic acids, which is called a codon, we'll kind of talk about it again. There are 3 nucleic acids, like if you have a U, U, and a C. Those 3 is what's a codon, and that code is for a specific amino acid. It's like saying if you have a UUC or something, I need an alanine amino acid. And then you have a CCT or something, that's for alanine. And then basically what you're doing is all these codons are code for a specific amino acid in a specific order. All right, and what the rRNA does is it provides the site for the assembling of these proteins. Okay, messenger RNA carries the information from the DNA to the ribosome. All right, and finally, transfer RNA brings the specific amino acids to the ribosomes for protein synthesis. Okay, so that's what the T does. It brings the amino acids. So how does it do it? So here they kind of are showing an RNA strand. It's a single strand. Okay, and you can see the amino acids. A, I think, is the alanine, and C, I don't remember what they all are. Okay, but the tRNA is drawn in this clover leaf shape. It's drawn like this because I guess that's kind of what it looks like in a way. Okay, with an acceptor stem at the 3 end, which carries the needed amino acids. These are the amino acids. I have 3 of them right here. Okay, and an anticodon which identifies the needed amino acid. That's what's called an anticodon. Okay, but basically the tRNA is what actually carries the amino acids over to the growing protein. Okay, so transcription is the synthesis of messenger RNA from DNA. Okay, remember, because transcription, the transcription, yeah, it brings the amino acid. Okay, so I'm sorry. So a transcription is the process where you take your DNA and it makes mRNA. And remember, mRNA is our messenger RNA that carries the information from the DNA to the ribosome. Okay. So the DNA splits into 2 strands, and the template strand, which is used in the synthesis of RNA, and the informational strand, which is not used. Okay. Transcription proceeds from the 3 end to the 5 end. And the transcription forms the mRNA with a complementary sequence to the DNA strand and an exact sequence as the informational DNA strand. So basically, it's doing the same thing as a transcription as we saw on DNA. However, it's doing it for the RNA, and instead of a C, it has a U, a uracil. And here you go. The difference between the RNA and the DNA is that you get a U in place of a T. Okay, so again, we've already seen this. You unwind the DNA and you have the genetic code. Okay, then you have RNA polymerase come by and do the exact opposite. Again, these nucleic acids are the exact opposite of the ones over here. Now notice I have a purple here because that's a U rather than here I have a T. Okay, and then you now have a new RNA strand. All right, which obviously we call mRNA. All right, and pretty much that goes into the ribosome and it does what it has to do. Okay, so if you want to get like the sequence, this is just kind of a problem. So if I have a C here, the daughter molecule has a G. If you have an A, you have a U. If you have a T, you have an A. If you have an A, you have an U, because remember, A and T's are on DNA. Okay, so it's always the opposite, and your strands and the order is the opposite. All right. Yeah, the informational strand is from our DNA. All right. So a sequence of 3 nucleotides, okay, a triplet codes, is for a specific amino acid. This is called a codon, okay? So the triplicate is called a codon. So for example, these 3 nucleic acids is for a specific amino acid, okay? For example, UAC is a codon for the amino acid serine, and UGC is a codon for cysteine. Codons are written from the 5' end to the 3' end of the messenger RNA. All right, so here just kind of gives you some examples. There are more than one order for a particular amino acid. So if you have all U's or UUC, those are for phenylalanine. Okay, so if you have a UA or UG, that's for leucine, and so forth and so on. We're not going to be responsible for that.